Hello, and welcome to a quick tutorial on fixing sequences for Invitrogen GeneArt Strings DNA fragments using the GeneArt Instant Designer tool. If you need help with importing sequences, please watch our separate video first for an example of those steps. Following import of sequences for a GeneArt Strings DNA fragments project, you may first see a pop-up window asking if you would like to optimize. This step is optional but can help fix any complexity that might make a sequence ineligible for submission. You can choose to optimize now, or you can review the details of your sequences first. Checking the order screen can help to summarize any issues with the sequences that need to be addressed. You can see in this example that 2 of the sequences have issues we'd like to review. Now, let's look at the Optimize tab. Here, the first sequence looks fine and can be ordered without any changes. This second item was uploaded as a protein sequence. In the process of converting the amino acid sequence to DNA, it has already been successfully optimized against the host organism. The third sequence does not currently qualify for synthesis as a GeneArt Strings DNA fragment. We can see why by clicking on Show Problems. It looks like there is a high GC region shown in the sequence. To address this, you must first select the host organism and define the ORF start and end. The number of nucleotides in this range must be a multiple of 3 to correctly identify the codons. Click Apply to show the protein sequence and enable the next steps. You may want to protect different aspects of your sequence prior to optimization. You can protect existing cloning sites by selecting from the list. This is a toggle to add or remove sites. You can protect regions if desired. Each of the selected sequences will not be changed during optimization. You can also define motifs to avoid in the final sequence, such as unique cloning sites or known regulatory sequences in your experimental system. You have 2 options for addressing the problems in your sequence. One is to fix problems, the other is to optimize the sequence. You can hover over the info icon to get more information. Fix applies a minimum number of required changes to meet GeneArt Strings DNA fragment criteria. Alternatively, you can fully optimize the sequence for the best chance of improved expression. For this example, we will select the minimal fix to qualify the sequence for strings synthesis. Fixing the sequence is successful. The sequence complexity was reduced so it can be built as a DNA fragment, while the encoded protein sequence has not changed. You can also view the quality graphs that provide information on codon quality before and after optimization. There are 3 different views: codon quality histogram, quality distribution, and GC content. You can see the GC spike that was moderated through codon substitution. Moving to the last sequence, we can see there are multiple regions with high GC content or repetition. Again, in the right-hand panel, the regions are marked along the sequence. In this example, the exact sequence is critical to the experiment, so we can decide to order the sequence as a clone instead of optimizing. To access this option, navigate to the Order tab. In the suggestions, we can select to order the sequence as a clone gene. This option allows the gene to be built and delivered in a production shuttle vector. You can then subclone the synthetic gene out of this vector and into your preferred final vector. All cloned genes are fully sequence verified. All that's left to do is add your items to the cart, where you can submit your order directly or generate an instant web quote. A web quote is an excellent way to ensure your custom DNA fragments are designed exactly how you want them before handing off to your purchaser.